just a minute. I'm starting. Okay, is my screen quite visible now? Is my screen visible? Yes.
It's visible, sir.
Okay, shall I proceed? I am audible as well.
Yeah, sir.
Okay, okay. ठीक है. So this semester I would be teaching you the practical aspect of this subject called metabolic engineering and systems biology. Okay, so actually what ideally should happen is that you should at least have some theoretical background before you start out with your practical. But anyhow, I thought I would not waste our time in getting you started out with your theory exam, because we would kind of have to get away with this entire 4 hours. So instead, what I thought was I will give you a kind of brief background on whatever is needed to perform our practicals, initial practicals, which are of course going to be very easy. And maybe you can start out with this practical, and in the meantime, when Dr. Vivek and Sucheta come and teach you the details of theory, then we can take some advanced level of practicals as well. Okay, so today what you can expect from this lecture is just a bird eye view of this subject, that is metabolic engineering. I will just give you some, give you some flavor of how things work in this subject. Okay, so the core of this subject, that is metabolic engineering and systems biology, is that you should now start thinking on manipulating at the system level rather than the component level. Okay, so well, say for example, for a time being, you might have learned how do you model, or you can say how do you prepare the model of a given structure of say certain enzyme, right? Using some modeling techniques like homology modeling. आता है या नहीं आप लोगों को? Right? Homology modeling करते आता है या नहीं आपको? Yes, are you guys there?
Where has, sir?
हाँ,
Could you
homology.
repeat?
Okay,
That
homology.
last part wasn't clear.
हाँ, it is going to be an interactive session, so no one is allowed to sleep. तो homology modeling में क्या करते हैं अपन? What do we do in
We
homology
look for
modeling?
homologous sequences and then align them.
No, no, I'm talking about what is the uh, what is what is the ultimate objective of doing that homology modeling?
We're assuming that having a similar structure will give a similar function.
Yes.
So we find
Hmm.
things having a similar structure.
Still, you are telling me what is the procedure. I am asking you, what do you achieve by that process? What is the end product of that process?
And
Huh?
as in,
As in, I mean, why do you do homology modeling? Yes, this is an open discussion. सिर्फ अपना देवकी ने ही आंसर देना चाहिए ऐसा नहीं है. There are other people sitting there as well.
you're using it to find structures of sequence of whatever you don't have a structure of before.
Exactly. The ultimate objective is that you create a kind of model for certain structures or for certain enzyme whose structure is not known, right? बरोबर आहे ना? That is the ultimate objective.
Yes,
That
sir.
means you have learned how to create a model of a given protein, right? That is what you know.
Mm.
Okay, so now do it happen ki like this protein would function in isolation in the cell? No, right? It would interact with some another components. Okay, it can if it is an enzyme, it is a part of say some metabolic pathway, right? So all in all, the thing is that it do not happen in vivo, or it do not happen in cells, that these single components work in isolation. Okay, the whole premise of life is that these chemical macromolecules perform a very orchestrated activity so that we generate a live cell. That is what the thing of life, the concept of life is. So at very basic biochemical level, what a cell is nothing but, you can say, an orchestrated you can say set of biochemical reactions that are happening, right? These reactions can be in terms of metabolic reactions, or these reactions can be in term of signaling pathways, and so on and so forth. So you can consider your cell as one of the system, and these system would have components, right? So so far so good. You have learned how to deal with small components. Now in this course and this course further, you would try to appreciate how these individual components would interact with each other, how would they talk with each other, and how that talk between the different component would enable the cell to perform certain function. Okay, and after understanding at such a system level, then you can think of how we can manipulate such systems for human purposes, or you can say for human benefits. Okay, so this is the very core concept of this metabolic engineering and systems biology. To understand the biological system, the biological you can say components like cells or the set of proteins on systems level, and to use this information for engineering, or you can say for manipulating or modifying those metabolic pathways, or those what you can say transcription system, or those signaling system, so that you can use them for the human interventions as such. So these human interventions can be in form of therapeutic opportunities, or they can be in form of industrial application, so on and so forth. Okay, so abhi I would just like to begin with what exactly do we do in this metabolic engineering stuff. ओके, तो पहले अपन शुरुआत किससे करेंगे अपन? वी वुड ट्राई टू अंडरस्टैंड व्हाट एग्जैक्टली हैपेंस इन दिस मेटाबॉलिक इंजीनियरिंग. ओके, सो नाउ कैन एनीवन जस्ट टेल मी व्हाट इज मेटाबॉलिज्म? मेटाबॉलिज्म क्या होता है?
Biological reaction happening within the body.
Okay, biological reaction that happens in cells, right? That includes metabolism. तो मेटाबॉलिज्म के दो मेन कंपोनेंट कौन से हैं?
Anabolism and catabolism.
Anabolism and catabolism. Okay, so whatever the components are, anabolism or catabolism, at their basic level, those are formed by biochemical reactions, right? Biochemical reactions, as you can see over here, we'll say for example, as you can see over here, metabolite A turning to metabolite B, or say for example, you can say that metabolite A through a biochemical reaction produces the metabolite B. Okay, and if such metabolic reactions, if they are consecutively arranged one after another, they form something called as metabolic pathways. तो metabolic pathways में क्या होगा? The product of one reaction forms the substrate of subsequent reaction. Okay, and such series of those reaction we collectively call it as metabolic pathway. Okay, now you guys are good at biochemistry than me, so I won't go in more details of these metabolic pathways. Okay, now can you just tell me how many types of metabolic pathways are present? मेटाबॉलिक पाथवेज के टाइप पता है आपको? यस, मेटाबॉलिक पाथवेज के टाइप पता है आप लोगों को? Oh, come on, guys. मेटाबॉलिक पाथवेज के टाइप है या नहीं लोग यहाँ पर?
है सर।
हाँ, तो गूगल सर्च कर रहे हो क्या? Okay, so let me tell you now कि there are different type of metabolic pathways. Metabolic pathways can be linear, right? Linear रह सकते हैं metabolic pathway wherein metabolite A is converted to metabolite B. This metabolite B in turn would be converted to metabolite C. Metabolite C would in turn convert to metabolite D, so on and so forth. This is, you can say, a linear form of metabolic pathways, right? Linear के साथ में they can be branched, right? Branch में क्या होगा? Well, say for example, in case of your glycolysis, your glucose would be phosphorylated to form glucose 6-phosphate. Then it would be isomerized into fructose 6-phosphate. Okay, and then there would be branching point, right? It would be split into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. बरोबर है? And it is possible that these two branch points can further lead to their own separate fates. बरोबर है? So this is included in something called as branch. Then there are circular metabolic pathways. जैसे कि for example, what are circular metabolic pathways?
TCA.
TCA cycle is a circular metabolic pathway. Can you give some another example?
Glycolate cycle. Urea cycle.
Urea cycle, right? Cori cycle. So these are all circular pathway. और एक है, can you imagine? Third type of pathway. Hmm? Okay, I will give you some time. इसके अलावा एक और पाथवे का टाइप है. ओके, आई विल गिव यू सम टाइम. नेक्स्ट लेक्चर में आप मुझे बताइएगा.
So the first type you stated was linear, within which you mentioned branch, and then
Then
you said cyclic, right?
cyclic. और तीसरा
Oh, would unbranched. The third thing.
unbranched would be in linear
Okay.
if it is not circular. Another type is something called as spiral pathway. आपको पता है स्पाइरल पाथवे का एग्जांपल? ओके, कैन यू इमेजिन कि भाई स्पाइरल पाथवे में क्या होता होगा?
Fatty acid oxidation, sir?
Yes, exactly. That means every time after following a set of biochemical reaction, your pathway would come at the same point, but the difference would be कि उसमें से एक कंपोनेंट कम हो चुका रहेगा, right? So in this way, spirally, right from the outside, it would come to the inside. So that is an example of spiral metabolic pathway. Okay, anyhow, whatever the metabolic pathway is, its core component, or you can say the heart of metabolic pathways, are metabolic reactions. Well, in a simple case over here, as you can see, you have got a simple metabolic reaction in which your metabolite A is converted to metabolite B, and usually these reactions are enzyme catalyzed reaction. Okay, मतलब क्या कि most of the biochemical reactions are catalyzed by enzymes. Okay, so now let us now view this biochemical reaction with some different perspective. Okay, so when we say that a metabolic reaction is happening in a cell, or that metabolic reaction is happening in your test tube, if you are doing some biochemical assays, what exactly is happening? That metabolite A is slowly being converted to metabolite B, or what you can in other simpler words say, the rate of change of concentration of A with respect to time. तो ये जो मेटाबॉलिक रिएक्शन मैंने यहाँ पर बताया है, in which your A is turning into B, how you can represent it? You can represent it in a statement like this reaction A to B is nothing but rate of change of concentration of metabolite A with respect to time. Or in other words, you can say that this metabolic reaction is nothing but rate of depletion of A with respect to time. Okay. So now just consider कि wherever you come across such expressions like rate of change of something with respect to time, you can easily reduce it in the mathematical form of a differential equation. तो जहां पर भी rate of change of something आता है with respect to time, such a kind of entities can be you can say easily reduced to a differential equation. So how we can reduce our metabolic reaction A being converted into B into a differential equation is in this way. So how do we write it? This d by dt correspond to what? D by dt is nothing but, how you should read it, rate of change of concentration of मेटाबोलाइट ए विथ रिस्पेक्ट टू टाइम. दैट इज ये वो जो नीचे का डिनोमिनेटर दिख रहा है dt, dt correspond to the change in time, while dA correspond to what? Change in concentration of A. So how do we represent it? Rate of change of A, that is dA by dt, is equal to Ka. That is, the rate of change of concentration of A is directly proportional. Okay, तो ये K जो है, well, it is the rate constant. Okay, so why do we have negative sign over here? ये negative sign क्या correspond करता है यहाँ पर? ये differential equation में,
because it's different.
yes, it indicates that the concentration of A is being decreased. Okay. Similarly, you can also say that this metabolic reaction can be represented as rate of built up of concentration of B with respect to time, right? And both of these things are somehow related with a rate constant. So ये A to B के conversion को what we can usually represent it as a set of you can say as a set of differential equations. Okay, so now this was in case of a simple A to B conversion or a simple A to B reaction. Okay, so now what happens is that this differential equation for multiple reaction that constitute a metabolic pathway thus can be linearly solved, right? तो metabolic pathway में क्या होता है? This A would be converted into B, this B would form the substrate for subsequent enzyme, and it might get converted into C, right? So in such a way, what you can do is that you can create differential equation for each and every subsequent metabolic reaction, and you can solve them using ordinary differential equations. So it is another mathematical method जिसे अपन क्या कहते हैं, ordinary differential equation or ODEs. So these ODEs can be used to simultaneously solve the differential equation that now correspond to a set of enzyme catalyzed reaction in your metabolic pathway. Okay, so now Dr. Vivek would be dealing with these things in more details, but I am just kind of trying to give you the very taste of how these things would work when you go in more details of this. For time being, आज के practical के लिए आपको इतना background काफी रहेगा. Okay, so what I have taught you today is that your metabolic reactions can be neatly reduced to set of differential equations. Okay, so now why would someone do such a thing? क्या जरूरत पड़ गई सीधी सिंपल मेटाबॉलिक पाथवे को कॉम्प्लिकेटेड डिफरेंशियल इक्वेशन में कन्वर्ट करने की? देयर मस्ट बी सम एप्लीकेशन विदाउट विच व्हाई वुड समवन बी इंटरेस्टेड इन डूइंग दैट? ओके, सो आई विल नाउ जस्ट गिव यू an application of where exactly this systems biology and metabolic engineering works. Okay, so I will give you an industrial application of this concept. तो अभी यहाँ पर क्या दिख रहा है आपको? You see a metabolic pathway over here, दिख रहा है? Okay, now can you just read it out and let me know कि ये कौन सा metabolic pathway रह सकता है?
This is glycolysis.
This is glycolysis. Okay, so now this particular metabolic model or this particular metabolic pathway was modeled in the same way as I have told you in the previous slide, and the ultimate objective of modeling this metabolic pathway was to kind of figure out how certain metabolites can be increased, or you can say how production of certain metabolites can be increased. तो देखो, you are advised to read this paper as I have shown over here. तो ये जो पेपर में इसका सारा how this system was modeled and how it was used is described in this particular paper. तो अभी यहाँ पर क्या दिख रहा है आपको? You can see that you have usual metabolic pathway, like you have got glucose, it gets converted to glucose 6 phosphate, then fructose 6 phosphate is produced, then you have got fructose 1,6 bis phosphate, then it is split into glyceraldehyde 3 phosphate and dihydroxyacetone phosphate, and eventually you end up with pyruvate. Okay, so pyruvate का आगे अल्टरनेट फेट्स होते हैं. Either it can go into TCA cycle, okay, in that pyruvate dehydrogenase reaction, or if it is or you can say if it faces say some anaerobic condition, it can undergo anaerobiosis and it can produce ethanol. Okay, it can have alternative fat and it can produce lactose. Okay, and you can see over here there are some alternative fat of this pyruvic acid. Okay, now one might think, okay, कि can once we model this system, okay, and once we model this system, then if we analyze this system properly, if we understand this system properly, then one can go on thinking like, how can we manipulate this pathway so that we want to increase this ethanol production? Okay, now as you know, ethanol is one of the very industrially active molecule, right? Ethanol का industry में बहुत ज़्यादा importance है. सो एंड इथेनॉल इज वन ऑफ द बाय प्रोडक्ट ऑफ ग्लूकोज मेटाबॉलिज्म, जैसे कि यहाँ पर अपने को इस मेटाबॉलिक पाथवे से दिख रहा है, राइट? सो नाउ वन कैन थिंक दैट इफ आई अंडरस्टैंड दिस मॉडल प्रॉपरली, then how can I manage or how can I manipulate this system so that I can increase the concentration of metabolite, this particular metabolite, that is ethanol. Similarly, you can also produce lactate, or you can also produce some another metabolite जो आपको इंडस्ट्रियली या फिर थेरेप्यूटिकली ज़्यादा रिलेवेंट लगता है, बरोबर है? So this is how things work. So the ultimate objective of this study was to create a model of this glycolytic pathway and its alternative fates, okay, and understand that metabolic pathway, understand that metabolic model, and then think of tweaking this metabolic pathway in such a way that you can force the flux or the flow of this carbon towards a desired end. Okay, so this is the very concept of utilizing this biological information for human welfare, right? So this is what we are going to learn in this due course. तो अपन आगे ये कोर्स में क्या पढ़ने वाले हैं? फर्स्ट वी वुड लर्न how do you construct a metabolic model, okay, in which we would first learn how do we generate the metabolic model first. Then I would let you know how do we obtain the rate laws, okay. Why? Because each and every metabolic pathway which contains metabolic reaction, and these metabolic reactions are catalyzed by enzymes, and these enzymes follow certain kinetics. अब काइनेटिक्स है मतलब क्या, उनको कोई रेट लॉ फॉलो करना है, ओके. यू माइट हैव derived the you can say Michaelis Menten equation, right, in your BScs, okay. That is one of the rate law that a particular enzyme might follow. तो ऐसे बहुत सारे रेट लॉज है जो अलग अलग एंजाइम्स फॉलो करती है डिपेंडिंग ऑन व्हाट काइनेटिक्स इट इज इन्वॉल्वड इन, ओके. सो अभी रेट लॉज है तो रेट लॉज के एक्सप्रेशन हो गए, रेट लॉस के एक्सप्रेशन है तो फिर उनमें काइनेटिक पैरामीटर्स हो गए, ओके, बिकॉज रेट लॉज आर मेड अप ऑफ kinetic parameters, and you need to figure out what are the initial values of those kinetic parameters so that you can efficiently construct a metabolic model. Okay, so this is what we are going to do in the very first step, right? You would learn how to construct a metabolic model. तो इसके तीन मेजर कंपोनेंट रहेंगे. हाउ डू वी जनरेट अ मेटाबॉलिक मॉडल, देन वंस यू जनरेट द मेटाबॉलिक मॉडल, अभी मेटाबॉलिक मॉडल जनरेशन में क्या क्या आएगा, यू वुड गो ऑन लिस्टिंग आउट what are the individual components of your metabolic pathway. In the previous case यहाँ पर कौन से component है, the components would be your metabolites like glucose, glucose 6 phosphate, fructose 6 phosphate, and all the constituent metabolites. First you would list out that, then you would list out what are the reactions that form those metabolic pathways, what are the enzymes that catalyze those biochemical reaction. In this way you would go on listing out what are small components that form your system, and eventually you would generate an effective metabolic model for a given set of biochemical reactions. Okay, so now you have listed out all the metabolites that are present in your pathway. Now you know what are the enzymes that are involved in the biocatalysis. Okay, now once you know the enzyme, it's time for you now to dig into the literature and to figure out what kind of kinetics those enzymes do follow, right? आपको हर एक एंजाइम का फिर क्या करना पड़ेगा, उसका रेट लॉ ढूंढना पड़ेगा. यू नीड टू फिगर आउट व्हाट काइनेटिक्स डू दैट एंजाइम्स फॉलो, and you can then further proceed to figure out what are the kinetic parameters once you know the rate loss, and eventually you need to figure out what are the initial values. तो ये सारी चीजें अगर आपके पास में होगे तो you can proceed for something called as kinetic modeling. क्या? समथिंग कॉल्ड एज काइनेटिक मॉडलिंग. तो काइनेटिक मॉडलिंग करने के लिए आपको क्या क्या पता रहना जरूरी होता है? व्हाट आर द कंपोनेंट्स ऑफ योर मेटाबॉलिक मॉडल, मतलब मेटाबोलाइट्स और एंजाइम्स. व्हाट इज द you can say what are the enzymes involved in, how do the enzymes behave, what are the kinetic parameters for its kinetics and their initial value. तो ये सारी चीजें अगर आपको पता होगी किसी मेटाबॉलिक पाथवे की, then you can perform kinetic modeling. Okay, so once you generate the metabolic model, then you usually perform something called as time course simulation. Okay, so time course, why do we perform this time course simulation? Usually the ultimate objective of performing time course simulation is to check out if your system reaches a steady state. Okay, now can you define what is a steady state? Steady state क्या होता है? You might have learned in biochemistry. What is steady state? Hello, are you there?
Yes, sir.
हाँ,
But I
तो
figured out the answer.
हाँ, क्या होता है स्टेडी स्टेट, क्या होता है बेटा, कोई बताएगा मुझे? या बताओ?
Sir, not.
नहीं पता. Steady state is when we say that a system has reached a steady state, when rate of formation of certain metabolite exactly equals to rate of depletion, right? At that state we say that the system has reached the steady state. Rate of formation equal to rate of consumption. We say that the system has reached the steady state. तो यहाँ पर mathematically acquiring the steady state will eventually ensure that the amount of metabolites that are influxed in your system are the same metabolites that are outfluxed. इसका मतलब क्या? जितने भी मेटाबोलाइट्स आपके सिस्टम में आते हैं उतने सारे स्टेडीली बाहर जाते हैं या नहीं. Well, that would be the ultimate objective of performing a time course simulation. कि भाई आपका जो सिस्टम है, whether it reaches the steady state or not, or if there are something called as metabolic leaks in your system. Okay, so the theoretical details of this time course and steady state and rate laws and everything, Dr. Vivek will deal with more details. तो अभी अपने को क्या सीखना है यहाँ पर कि सबसे पहले अपने को क्या करना पड़ेगा? Practical point of view, we would need to generate a or construct a metabolic model. Then we will have to make sure that whatever model that you have generated, yes, of course, it obtains a steady state. Okay, so once you make sure that your system reaches the steady state, then you can further move on to perform some higher state of analysis like metabolic control analysis. Okay, तो metabolic control analysis का ultimate objective अपने practical में क्या रहेगा? It would be to obtain the flux control coefficients. Okay, so I would just tell you in more details in my next slide ki what exactly is this metabolic control analysis and what exactly are the flux control coefficient and everything. Okay, so once you perform this metabolic control analysis, then you would obtain the flux control coefficient, and once you obtain the flux control coefficient, then you can fine tune the model to direct the flux of the metabolites to the desired effect. Okay, so just to let you know, I would show you graphically how things happen over here. So this figure is of course the one that you have already seen. This is how we generate a metabolic model. then what we do is that we obtain the rate laws of the enzyme. अभी यहाँ पर क्या क्या दिख रहा है आपको? lactate dehydrogenase दिख रहा, which is somewhere over here. So these enzymes will follow this rate law. Okay, then you have got this PDH enzyme. Okay, PDH enzyme कहाँ पर दिख रहा है आपको? Can you tell me? This is the enzyme number 14 in your metabolic model is PDH, and it would follow this rate law. Okay, then you have got this enzyme PTA जो यहाँ पर दिख रहा है, reaction number 15. It would follow this particular rate law. Then you have got this ACK, that is reaction number 16. It will follow this particular rate law. So what you need to do is that you need to figure out how this enzyme behaves mathematically by obtaining the rate laws. Okay, you can obtain such information about the kinetics and rate laws by by सर्चिंग डेटाबेसेस लाइक ब्रेंडा एंजाइम. ये जो डेटाबेसेस है इसमें आपको मैक्सिमम एंजाइम्स का काइनेटिक्स और रेट लॉस के बारे में आपको ज़्यादातर इंफॉर्मेशन मिल ही जाएगा. Okay, for in our case, for the sake of simplicity, we have already used a system whose rate laws are already known. Okay, so you don't have to deal with this stuff initially. At least I will tell you what are the rate laws that these enzymes might be following. Okay, उसके बाद में as you can see over here, the next thing would be to obtain the kinetic parameters and their initial values. Okay, so well, now again let us stick on to the first rate law for the enzyme lactate dehydrogenase जो यहाँ पर दिख रहा है LDH. Okay, तो इसके अंदर क्या था? It follows some rate law, and this rate law में क्या होते हैं? There are kinetic parameters in your rate law. Well, say for example, this LDH enzyme has got kinetic parameter like V, then constant for pyruvate, constant for NADH, this KEQ, KLAC, KNAD, so on and so forth. So collectively, this kinetic parameter would form a rate law, and this rate law would eventually decide how your LDH might function, or at least mathematically वो कैसे behave करेगा. Okay, so in this way you have then enzymes, their kinetic parameters. Okay, and you need to figure out what are their initial their values, of course, and their initial concentration. Okay, बात समझ में आ रही है आपको? उसके बाद में once you have everything in place, what you can do is that you can then perform time course simulation in which you would get such graphs जैसे यहाँ पर दिख रहे हैं आपको. Okay, and if such graphs then converge यानी क्या कि ऐसा when they reach a plateau, that means the steady state has reached. Okay, so once you make sure that the steady state has reached, you would perform something called as metabolic control analysis. The objective of performing metabolic control analysis would be to obtain the flux control coefficient. तो यहाँ पर as you can see, the flux control coefficient of these reactions are shown with these numbers. तो यहाँ पर you can say, for the sake of simplicity, you can consider it as कि the flux control coefficient of say certain enzyme would indicate how much mass flow is happening through that particular reaction. Okay, so flux control coefficient are hello.
So are you on the third slide or is our screen frozen? We're a bit unsure on that.
Do you see this MCA?
On the third slide?
Do you see this metabolic pathway first? Objectives क्या दिख रहा है आपको?
Industrial application, and there's an image of the pathway.
Objectives वाला slide नहीं आया आपको?
No.
नहीं?
नहीं.
सबका वैसे ही हो रहा है because I have moved 2 slide ahead.
Okay then. I guess it wasn't for us.
Okay, सबको वैसे ही हो रहा है क्या? या सिर्फ क्लास में देव की बैठे वाली है?
Yes, sir.
हाँ?
Sir, we are able to see third slide.
Okay, maybe then I will stop my sharing, and again I will share it. Okay, just a minute. ओके, अभी दिख रहा है? हेलो,
But I still see the third
तो
slide.
अभी नेक्स्ट स्लाइड दिख रहा है?
Third.
नेक्स्ट स्लाइड दिख रहा है आपको?
नहीं.
नहीं सर.
I am moving forward now. You should see it. अभी भी नहीं दिख रहा है?
नहीं sir.
No, sir.
Okay, then let us wait for a while then. अभी क्या दिख रहा है?
Now we see the fifth slide, sir.
Okay.
And sir, Mohit was asking you to check the chat box as well. I think his speaker is not working.
Okay, now I am already recording this thing, huh? So this
Okay.
would happen that I will give him the recording stuff so that he can check it out. Okay, तो सबसे पहले क्या करते हैं अपन? We generate such metabolic model, then we obtain the rate laws. Once we obtain the rate laws, then we obtain the kinetic parameters जो यहाँ पर दिख रहे हैं आपको. Okay, once we obtain the kinetic parameters, we perform time course simulation, and then once we are sure that our enzyme or that our system reaches the steady state, then we proceed for finding out the flux control coefficients. Okay, तो flux control coefficients क्या बताता है आपको? It would tell you that around a branch point how the carbon flow might work. Okay, so well, say for example यहाँ पर in this case दिख रहा है आपको यहाँ पर number 5 MCA FCC.
Yes, sir.
Okay, so this particular figure represent that the flow of carbon from these two metabolites when it reaches to pyruvate, it is 1.97. Okay, and at this branch point it gets split into either towards lactate and some another fate. तो ये distribution कैसा होता है? At the rate of 1.12 and 0.81. So these numbers would give you an idea on how, or you can say at the branch point, how the carbon flow would be. Okay, so now once you have those number, what you can do is that you can then fine tune your model so that you can adjust the flow of metabolites in a desired direction. ठीक है, so the same figure is represented over here now. दिख रहा है यहाँ पर? This is what pyruvate at the branching point. Pyruvate के branching point में lactate की तरफ कितना कार्बन फ्लो हो रहा था? At the rate of 1.2. और pyruvate की तरफ कितना जा रहा था? At the rate of 0.8. So this would give you an idea how the carbon flow would be in your metabolic pathway towards the different fates. So now what you can do is that you can fine tune such a such a flux control coefficient by adjusting such flux control coefficient. What you can do, as you can see over here, you can adjust the flow towards a desired direction, so that जो पहले यहाँ पर flow PEP से pyruvate through ACCOA की तरफ जा रहा था, now the same flow would be diverted towards a desired product, that is ethanol or lactate. So that is how we do the fine tuning, and again simulate your metabolic pathway and see if the if the desired results has been obtained or not. Okay, so this would give you a clue on what enzymes are to be modulated in what way so that you would get a desired end product. Okay, so this was just an example of how this metabolic engineering is used in systems biology. Okay, so ये होने के बाद में once you finish out with modeling and manipulating this enzyme sorry this metabolic pathway, then you would perform something called as mini project. अपने पास में mini project भी है, और ये mini project में we would deal with some therapeutic application of systems biology. Okay, so in this mini project what we will do is that we would try to simulate your polyol pathway in diabetic conditions. ओके, तो कोई बताएगा कि डायबिटीज में पॉलीऑल पाथवे कितने लोग थे बायोलॉजी ऑफ डिसीजेस में? हेलो, देवकी नहीं थी फॉर
Not
श्योर.
me, sir. I wasn't, sir.
She wasn't there. तो it is responsibility of others who were there in that course जो बताएंगे कि डायबिटीज में पॉलीऑल पाथवे का क्या रोल होता है. कौन बताएगा? नहीं, अरे कैसे रे, कैसे होगा ऐसे से? ठीक है, एनी हाउ मैं बताता हूँ आपको. सो डायबिटीज में क्या होता है? यस ऑफ कोर्स इट एवरीथिंग स्टार्ट्स आउट विथ एक्सेस ऑफ ग्लूकोज. तो द थिंग इज कि इन नॉर्मल शुगर होम्योस्टेसिस जब डायबिटिक कंडीशन नहीं होता है, यूजुअली योर ग्लूकोज एंटर्स इनटू दिस ग्लाइकोलिटिक पाथवे. ओके, और ग्लूकोज का आपका जो लेंस होता है, you say eye lenses, they have got some alternative fate of glucose over there. That is, this glucose is also converted into sorbitol. Okay, so अभी ग्लूकोज के दो अल्टरनेटिव फेट्स है in your lenses as such. Okay, either it can go into glycolytic pathway or either it can go into sorbitol. So a delicate balance is usually maintained in normal sugar homeostasis, is that about only 3% of the glucose would be diverted toward formation of sorbitol, and the remaining 97% would be diverted towards this glycolytic pathway. Okay, so now as you know that during diabetes this glucose metabolism is somehow hampered. Okay, and in such diabetic condition, instead of only 3% of the glucose that is to be sent toward formation of sorbitol, well, due to you can say due to inefficiency of glucose to get entry into glycolytic pathway, 10-fold of glucose is now diverted toward formation of sorbitol. तो मतलब यहाँ पर 3 की जगह पर कितना परसेंट ग्लूकोज अंदर जाएगा sorbitol की तरफ? 30%.
30.
Okay, so now what happens is that this glucose would be converted to sorbitol using the enzyme called as aldose reductase. This sorbitol would be acted upon by sorbitol dehydrogenase to yield fructose, and this fructose using hexokinase, that is one of its phosphorylation event, it would regenerate this fructose 6 phosphate, and this fructose 6 phosphate would further enter either into glycolytic pathway or it can go towards glucosamine 6 phosphate synthesis. Okay, so the delicate balance is usually disturbed in such diabetic condition. So the pathway in which glucose is converted to sorbitol, the sorbitol is converted to fructose, is called as polyol pathway. Okay, तो डायबिटिक कंडीशन में क्या हो जाएगा? पॉलीऑल पाथवे का जो फंक्शनिंग है वो बहुत ज़्यादा अमाउंट में होता रहेगा. वेल नाउ द प्रॉब्लम इज कि दिस aldose reductase enzyme is highly effective in its catalytic turnover, but that is not the case with this sorbitol dehydrogenase. तो ये जो सॉर्बिटॉल डिहाइड्रोजिनेज है इसका के कैट बहुत कम होता है एज कंपेयर टू अर्लोज रिडक्टेज. इट इज नॉट वेरी इफिशियंट ओके इन इट्स कैटालिसिस. सो होगा क्या, होगा यू कि ड्यू टू दिस रीजन this sorbitol would now somehow accumulate in your lens tissues. Okay, बात समझ में आ रही है आपको? Sorbitol would accumulate. Now sorbitol being osmotically active, it would start to attract water. इसकी वजह से आपका जो लेंस टिश्यू है वहां पर क्या हो जाएगा, ड्यू टू अक्यूमुलेशन ऑफ सॉर्बिटॉल इट वुड क्रिएट ऑस्मोटिक प्रेशर एंड सेल वुड बस्ट. और सेल बस्टिंग की वजह से there would be you can say cloudiness in your lens, and we say that a person has developed cataract. तो अभी why don't we generate this metabolic model? Okay, and the story don't end here. It would also disturb the balance of NAD and NADH that would eventually result in formation of reactive oxygen species. ओके, तो इससे रिएक्टिव ऑक्सीजन स्पीशीज भी एक्सेस अमाउंट में तैयार हो गए. ठीक है, रिएक्टिव ऑक्सीजन स्पीशीज यानी क्या, हाइड्रोक्सी फ्री रेडिकल, ओके, सुपर ऑक्साइड रेडिकल्स. well these are all dependent on this NAD to NADH reaction. okay, so रिएक्टिव ऑक्सीजन स्पीशीज यहाँ पर ज्यादा अमाउंट में प्रोड्यूस होंगे, सॉर्बिटॉल ज्यादा अमाउंट में प्रोड्यूस होगा. सिंस इट इज ऑस्मोटिकली एक्टिव इट वुड क्रिएट ऑस्मोटिक प्रेशर, the cells would burst, it would create cloudiness in the lens, and eventually cataracts are developed. Okay, so now this is the very premise of our mini project. Okay, तो ये mini project में अपन क्या करेंगे, we would try to generate a metabolic model for this system, and we would simulate our metabolic model in two ways. First, only 3% of the glucose flow we would let it allowed to go towards sorbitol, and the remaining in glycolysis, and check out what happens to the reactive oxygen species as well as sorbitol. The next thing that we would do is in diabetic condition wherein we would increase the flux by 10 fold, that is 30% would be allowed to go through this way and the remaining would be allowed to go this way, and again we would check what happens to the reactive oxygen species concentration as well as sorbitol. So in this way it would kind of help you understand the delicate balance of how ROS are produced, how sorbitol gets accumulated, and then we can think of performing metabolic control analysis to figure out how the flow of this glucose can be manipulated so that most of the glucose can be diverted away from sorbitol so that this reactive oxygen species can be controlled, okay, or the production of sorbitol can be minimized. So ये सारी चीजें अपन क्या करने वाले हैं, we are going to deal with in the entire semester. Okay, so this is what I have planned for you this year. Okay, so पहले अपन क्या करेंगे, I would give you a small assignment today. Okay, तो ये assignment कुछ ऐसा है कि आपको क्या करना है, we would begin with something you already know. What you are supposed to do is that you are supposed to write a program that calculates the velocity of an enzyme catalyzed reaction over specified time. Okay, so okay, so now do you remember the basic biochemistry course? If you plot velocity against substrate concentration, आपको कैसा मिलना चाहिए प्लॉट, आपको प्लॉट कैसा मिलना चाहिए, समथिंग लाइक दिस, राइट? ये प्लॉट क्या बताता है?
Wait, sir, are you on the 5th slide?
Yes, of course. दिख नहीं रहा है?
Okay. I'm just confirming.
मैंने बनाया वाला प्लॉट नहीं दिख रहा है क्या आपको?
No, sir, I don't see any plot.
Yes, बाकी के?
No, sir.
Okay.
Sir, is it a graph below the kinetic parameters and their initial values? Or is it something else?
Just a minute. Okay, I will again stop the sharing. Okay, I will again show you. अभी दिख रहा है? Can you see now assignment?
Yes, sir.
Okay, now can you see what I am drawing now? Wait, I have started. अभी दिख रहा है मैं क्या ड्रॉ कर रहा हूँ?
No.
नहीं?
No.
Okay, do you remember the Michaelis-Menten kinetics? Michaelis-Menten kinetics में क्या करते हैं अपन?
Enzyme and substrate concentrations.
We plot the substrate concentration on x-axis and velocity on y-axis. Okay, so as the time progresses, the substrate concentration, the velocity increases, right? The velocity increases, okay, and the velocity would reach at certain point, okay, and we say that this is the maximum velocity, and the substrate concentration where half of the maximum velocity is reached, okay, उसे क्या कहते हैं अपन? We call that substrate concentration as Km, right? So अभी आपको क्या करना है? You need to write a computer program that would follow the consider any enzyme, or consider any metabolic reaction that would follow the metabolic conversion of A to B maybe, and it follows the Michaelis-Menten kinetics. आपको क्या करना है? You need to write a computer program जो क्या simulate करेगा? It would simulate how this enzyme would behave with the given time. Okay? तो इसमें होगा यू कि what you would ask is all the parameters of your Michaelis-Menten equation. Michaelis-Menten equation क्या कौन सा है? What is the equation of Michaelis-Menten equation? Velocity is equal to Vmax okay into substrate concentration upon Km plus substrate concentration. Okay, so you need to write a computer program that would take all these parameters into itself, okay, and calculate the velocity of an enzyme catalyzed reaction, okay, and how this enzyme would behave with time. Okay, this is what you need to do. Okay, now I will stop
So,
these things.
how it behaves with time in the sense, are we assuming that the substrate concentration
Would
increases
decrease.
or decreases in a certain manner?
Yes, of course. Well, देखो आपने को क्या करना है, ये जो Michaelis-Menten इक्वेशन वाला एंजाइम है, इसका इसका अपने को सिमुलेशन करना है कि भाई एट इनिशियल कॉन्सेंट्रेशन ऑफ सबस्ट्रेट इसका वेलोसिटी कितना रहेगा, राइट. सम ऑफ द सबस्ट्रेट वुड बी कंज्यूम एंड वुड बी कन्वर्टेड इनटू प्रोडक्ट एट सेकंड 1. एट नेक्स्ट स्टेप, एट नेक्स्ट सेकंड व्हाट वुड हैपन, द बचे बचे वाला जो कुछ भी सबस्ट्रेट रहेगा उसमें से कुछ यूटिलाइज होगा, प्रोडक्ट में कन्वर्ट होगा.
Okay.
Okay, and then you would be plotting the velocity, right?
Okay.
ठीक है. So now how would you correlate your enzyme kinetics with time? क्योंकि हर एक स्टेप के बाद में क्या होना चाहिए, सबस्ट्रेट का कॉन्सेंट्रेशन कम होना चाहिए,
Yes, sir.
राइट. सबस्ट्रेट का कॉन्सेंट्रेशन कम होना चाहिए. सॉरी, यहाँ पर क्या है, टाइम है यहाँ पर. सबस्ट्रेट का कॉन्सेंट्रेशन कम होना चाहिए. तो हाउ वुड यू कोरिलेट इट विद, ये कैसे क्या आप मॉनिटर करोगे?
So we update the substrate concentration
How?
after every step.
Every step, how would you do that?
We're assuming you have used
तो कुछ ना कुछ तो माइनस करना पड़ेगा ना. तो क्या माइनस करोगे?
the amount of substrate
देखो, the
consumed.
thing is, you can correlate it like the amount of substrate used would be dependent on the velocity of an enzyme.
Yes.
Okay, तो V का जो कुछ भी कंपोनेंट रहेगा, if you subtract it from the substrate, okay, बात समझ में आ रही क्या आपको?
Yes, sir.
Your substrate concentration is equal to substrate concentration minus velocity. ये हर बार आपको इसमें से माइनस करना पड़ेगा.
Okay, sir.
At every time step. Okay. That would be your new substrate concentration, and then check out if you get such curve or not. Okay. दूसरा प्रोग्राम आपको ऐसा लिखना है जिसमें you would save the velocity as well as substrate concentration. पहले इसमें आप वेलोसिटी प्लॉट करेंगे और दूसरे ग्राफ में यू वुड प्लॉट सबस्ट्रेट कॉन्सेंट्रेशन एज वेल. तो सबस्ट्रेट कॉन्सेंट्रेशन डिक्रीज होगा और वेलोसिटी इंक्रीज होगी, राइट?
Yes, sir.
Okay, so this is what you are supposed to do in this week. The next week में we would go towards certain different thing. तो अभी मैं क्या करूँगा, I would share this video with you guys, okay, and maybe you can see this video again and try to understand कि exactly आपको क्या करना है. हाँ, ठीक है.
Sir, are there any values we should assume?
नहीं, दैट देखो, यू नीड टू फिगर आउट ये अच्छा ग्राफ कब आएगा. द वन दैट यू सी इन योर बुक इज दैट व्हेन यू गिव प्रॉपर वैल्यूज टू दीज पैरामीटर्स. ओके, तो आपको थोड़ा सा लिटरेचर सर्वे करना पड़ेगा. टेक सम एंजाइम, एनी एंजाइम विच फॉलोज दैट माइकलिस मेंटेन काइनेटिक्स, ओके, और उसका एक्सपेरिमेंटल वैल्यू ढूंढ कर निकालो एंड ट्राई टू फिट दोज वैल्यूज इन योर मॉडल.
Okay, sir.
ओके, ठीक है. ओके, सो आई एक्सपेक्ट यू कि अगली बार आप मुझे वो चीज़ ईमेल कर देंगे. ओके, तो दिस इज हाउ थिंग्स वर्क्स. ओके, तो इसके बाद में we would try to see how the effect of inhibitor is. Then we would try to write the codes in which we can extend this pathway to include another reaction, so on and so forth. ठीक है, कॉम्पिटिटिव अनकॉम्पिटिटिव वो भी करके देखेंगे अपन, इफ वी गेट द सेम टाइप ऑफ ग्राफ्स और नॉट. हाँ, तो ये सारी चीजें पहले अपन करेंगे सो दैट योर थ्योरी वुड बी कवर्ड अप प्रॉपरली, एंड देन वी वुड गो ऑन यूजिंग द सॉफ्टवेयर. हाँ, तो वी वुड बी यूजिंग द सॉफ्टवेयर कॉल्ड एज कोपासी. ओके, तो अपन कौन सा सॉफ्टवेयर यूज़ करेंगे? कोपासी सॉफ्टवेयर यूज़ करेंगे ये मॉडल जनरेट करने के लिए। और वेल, आई एम टॉकिंग अबाउट these मॉडल्स, दिस एंड दिस मॉडल। ओके, तो ये दो मॉडल जनरेट करने के लिए वी वुड बी यूजिंग कोपासी। और फिलहाल आप ये छोटे मोटे कंप्यूटर प्रोग्राम्स लिखना शुरू कर दो। ओके, विल इट वर्क?
Sir, any specific language or?
नहीं नहीं, वॉट एवर यू विश, कोई कोई दिक्कत नहीं है मुझे,
Okay.
ठीक है, बट डोंट यूज फोर्ट्रेन और ठीक है, you can use java, you can use perl, you can use python, whatever you are comfortable with.
Okay, sir.
ओके, ऐसी कोई लैंग्वेज यूज मत करो जिसके कंपाइलर ही ना मिले।
Sure.
ठीक है, सो इट शुड बी काइंड ऑफ कन्वीनियंट फॉर मी एज वेल टू इवेल्युएट योर असाइनमेंट्स।
Okay, sir. Got it.
ओके, तो आई विल स्टॉप this recording now. Okay, and I will share this recording with you, right?